my ceiling.
It kind of is. All right. Now you're in twice, and
Hello.
Hello.
Hello. Can you hear us?
Yes. Hello. Hello.
Hi, Ken.
Hi. How are you?
They've logged in twice,
Hang on a
right?
second.
There's two of them, one for the presentation and one for the voice.
What?
that's right.
Okay. Hi, Ken. Can you hear me?
Yes.
Yep.
Good.
Okay, so we're going to use the phone for audio and the computer for
Hi,
the
Ken.
visual
Can
feed.
you see
Is
me?
that it?
Can you see us?
Hello?
Hello. You have an audience of 2.
Okay.
3. An audience of 3.
Hello?
You can't see us. You can't see us.
The top left.
All right, can you
On
see
the
me?
top left. See, look on the top left.
But he'll see it as his main screen.
Yeah. All right, that's top right.
I like the new flags.
All right, how do we get the
He's got the flags
computer
from
in?
Tepos Lawn up in his house.
Yeah.
All right,
Hey,
so how
Ken.
do we get the
Go to
Ken,
Zoom.
can you hear
and
us?
share.
right here?
Yes, I can hear you.
It says
Click
click if you
here.
want to switch to a different microphone or speaker.
I don't know if he can hear us.
It says click if you want to switch.
Ken, can you hear us?
I can hear you.
Okay, great.
I like the Tepos Lawn flags in your loft. Nice look.
Uh-huh. You didn't know that?
I haven't been to your loft since then.
Yeah. Let me put you on speakerphone here, I think.
Can you see us
Hello?
here? Can you see us here?
Yeah, okay. All right, now let's go to...
It's fine. He's in control.
I'm trying to get
Got
it to
it.
broadcast.
Uh-huh. It is, isn't it broadcasting? No?
Yeah, we hear you. We hear you and see you.
We're gonna start.
Right, but you don't see my presentation, do you?
Yeah, we do. But you need to put it into presentation.
Put it
mode.
into full screen.
Yeah, put it in full screen exactly. And go to the beginning, I guess. At the moment, it looks like it's kind of third way through.
Yeah. But now that would show up here.
That's it.
Oh, there.
Do you mean to start there?
What?
This is the presentation. I hope you got all that.
Yeah, like, are you going to go to the start?
Okay.
Okay, you're going up.
Are we good?
Okay, we can start. You're not on the start of your presentation at the moment.
I believe I am. Yeah, here we go. Yeah.
Okay, all right.
This is where I'm starting.
Okay, all right, fine. So everybody, Dr. Ken Alper.
Hello? All right. Can everyone hear me?
Yep.
Yeah, that's it.
Okay. All right. So I was, for this presentation, I'm recalling a lot of conversations I've had with people regarding mechanisms of action and so on, in which I wish I had uninterrupted time,
Sorry,
you
Ken,
know, a
can
captive audience,
can you move a little
and
bit?
my PowerPoint in front of me. So I'm taking advantage of that. So basically, what this is going to be is a set of comments regarding mechanisms of action that reflect a lot of recent conversations that I've had regarding this topic. We start with Ibogaine and its structural derivatives. Catharanthine is a particularly interesting one. Catharanthine is the mount. Are you guys? Do we need the video of me, or are we just looking at the screen?
We can see you.
Looking at you.
Can you?
Yeah,
move-
because my clamp that holds the camera is misbehaving. Let's see.
Can you move a little bit further away from the microphone?
Okay. How's that? Can you hear me?
Further away from the mic.
I should get further away from the mic.
Yeah, because there's a limiter on it.
Oh, okay. I'm clipping, huh?
Yep.
I'm okay now?
That's better.
Okay. All right. So, and everybody can see the screen. Okay. So, Ibogaine has a structural analog, an interesting one called catharanthine, which is in the lower portion of the of this figure. Catharanthine is part of a bisindole alkaloid, or a couple of bisindole alkaloids called vincristine and vinblastine. They're used in cancer chemotherapy. So there is a tremendous economic interest in studying how catharanthine is produced. And recently, we were able to get one of the catharanthine researchers, Sarah O'Connor, interested in Ibogaine, and she has recently published a paper on how Ibogaine is produced in the T. iboga plant, and Gita was instrumental in getting her. Well, hello.
Yeah, we can hear you.
Okay, so Gita was instrumental in getting her the material, plant material that she needed to work with. So that is a concrete contribution, the Journal of Biological Chemistry last month of Gita's work. And it's interesting that catharanthine is so well studied, and the person who was studying it was not aware of its structural similarity to Ibogaine until we had contacted her. Okay, so we always start with the history, and the history really starts here in the mid 50s. With a patent for the use of Ibogaine to potentiate opioid analgesia. In other words, to potentiate the effects of opioid analgesia and offset tolerance by an unknown mechanism of action. This patent expired many decades ago, but one wonders if there were a compound these days that was rolling back tolerance to analgesic medication, it might be of interest to the industry, particularly if it represented a novel mechanism of action. But evidently, this clue was overlooked and remains a footnote to history. Howard Lotsoff was aware, I think, of this work because he knew that Ibogaine had been classified as a hallucinogen. And in the early 60s, he and a number of people were lay drug experimenters and engaged in the research methodology of that day, which is basically to take the substance and take notes on their experiences. Eventually, some of them that were heroin dependent got a hold of Ibogaine, including Howard, and were serendipitously detoxified. These are some. This is an image from early treatments that were done in the Netherlands in the late 80s, early 90s. They were done in this apartment in which Howard and Norma are standing in front of. Here to Franken is a figurative artist who was also part of the scene. Importantly, this is where the nexus of harm reduction Ibogaine begins. Nico Adriaens was active in a network of Dutch heroin users that were involved in these Ibogaine treatments. Adriaens is a seminal figure in the history of harm reduction. He founded the Rotterdam Junkiebund, which is the first drug users union, and drug users unions remain influential in EU drug policy these days. The Junkiebund originated a number of harm reduction methodologies, including needle exchange, which was actually, and this is before AIDS. The needle exchange was to help the spread of hepatitis B. And there was a strong identification among these squatters, heroin users, with IV drug use. And there's always been a nexus of opioid dependence in Ibogaine. That has always been the indication for which Ibogaine is most specifically seems to have been used. This is an iconic image from the Ibogaine subculture painted by the figurative artist who was standing with Howard and Norma in front of her apartment. And one interesting panel here, a representation of the spirit within heroin, which I've always found to be evocative. Interestingly, in the middle panel, we have Apasmara, the demon of ignorance, being subjugated by Shiva. And notice that the middle panel of the triptych, where the demon of dope is being subjugated by the artist, this is replicating the Shiva image. It's also interesting because Shiva is a favorite household god of untouchables, who are the lowest status of the Hindu caste system. And that's a hierarchy that's delineated basically by the capacity for ritual contamination. Death is highly contaminating, and accordingly, labor viewed as appropriate for untouchables includes tasks that involve contact with the dead. Shiva visits burial grounds, interacts with the spirits of the dead and their cremated ashes to his own body, and is not contaminated by this. This is all, I think, very wheaty. Intravenous junk users, like untouchables, are highly stigmatized and viewed as lowly and contaminating. And there is a process of decontamination that I think is implicit in recovery. Howard and Norma in front of the FDA building. This is part of Howard's efforts to have Ibogaine developed in the mainstream, and he had significant progress towards this. For one thing, there was also an activism behind this, as well as Howard's diplomacy. And here we have kind of a famous incident where some activists capitalized on an action that had been taken place at the NIH a couple months earlier. On the right was a huge action that had been organized by Larry Klamer and ACT UP to storm the NIH for the development of AIDS medication, which, by the way, proved to be tremendously successful. A few months later, a few dozen people passed out these leave lists and managed to probably convince NIDA that another action was imminent, because NIDA did go ahead with its Ibogaine project at that time. So between 1991 and 96, NIDA supported preclinical contract work, including toxicology and pharmacokinetics, which enabled a phase 1 study of a single dosage of Ibogaine and cocaine dependence. That study was approved. Humans did receive Ibogaine in a clinical study in the US starting in 1993. They probably got up to a dosage level of 2 milligrams per kilogram before the study was terminated by intellectual property and contract disputes. There were no safety issues that were reported in this trial at these dosages. And there's been no subsequent research done in the US since the NIDA Ibogaine project. Eventually, NIDA terminated its Ibogaine project, and simply because they decided that they did not have the resources to proceed with the study on their own, and also they lacked the human data from a phase 1 study that should have been produced. Instead, the study had fallen apart. So that was the end of the first NIDA Ibogaine project. But this is summarizes a lot of this work here. This is a one-figure summary of all of the preclinical animal model work that's been done with Ibogaine on the self-administration of either morphine, alcohol, or cocaine. Now here, you can see my pointer. Yeah, okay, this comes across. Okay, this midline, 000, would be no effect. And everything to the left of the effect is reduced self-administration. Everything to the right of the no effect level is an increase in self-administration. So look at the consistency of the reduction across all of these studies. And this is the overall effect here. Okay, so basically, if you combine this with animal studies and the animal models of withdrawal, there is an animal model of withdrawal. You get the animal model, animal dependent on morphine, and then you give naloxone to precipitate sudden withdrawal. And if Ibogaine is on board, that greatly diminishes withdrawal symptoms. That has been done 14 times with 13 positive results. So if you add all of this together, there's something like 50 plus animal studies showing either diminution of withdrawal or diminution of self-administration with Ibogaine. Okay, so following the NIDA Ibogaine project was the NIDA 18MC project. That began in 2013. A total of $6 million in modern day costs has been expended, approximately equivalent to the $2.5 million that was spent on the NIDA Ibogaine project in the early 90s, if you adjust for inflation. And basically, it's reached a similar point. 18MC is ready to be given by humans, and they're There is the clinical trial has been enabled in a regulatory sense, but there's no funding for it going forward. 18MC, by the way, is a putatively safer analog of Ibogaine, developed by a process of rational synthesis, which means that the uh, the the developers were purposely systematically using their knowledge of structure activity relationships to develop a safer congener. As you can see, 18MC and Ibogaine only differ at these positions, and this is the core iboga alkaloid structure. 18MC appears to be less toxic in Ibogaine, particularly with regard to herd blockade and experimental evidence that would model the liability towards arrhythmia. Humans have taken in 18MC outside of the US in phase 1 studies, but a phase 1/2 study, which would be the next step here, has yet to be funded. So what's followed in the absence of any kind of official approval is the Ibogaine medical subculture of people using this in various settings, such as hotel rooms and clinics outside of the United States. Just a little bit about the public health significance of the opioid crisis, which is really not the main emphasis here, but there was a few slides that really stood out. I teach a course to the grad students at NYU on addiction, and every year I have to update my slides, and it's amazing how every year there is a major update to do because there are so many, the volume of developments in that crisis is so great. But here's a couple images that, I often throw out there. This is a meta-analysis of studies of looking at mortality following populations of drug users for cumulative deaths over time. So this is a meta-analysis. If you're adding all of these points or studies, and basically, if you put all this information together, you can generally say that opioid use cuts the lifespan in half. The lifespan is expected lifespan is usually mid-late to late 30s, with over half of the premature accounted for by overdose. Other accidents and illnesses account for a lot of the other mortality as well. And this is older figures, and probably the newer figures are even worse because, for one thing, we now have fentanyl. Now, this is the curve. Those of us, let's see, the NYU Ibogaine conference was back here, like in the year 2000. This is about the time that I was getting seriously involved in it. And now we're here in 2016, and you can see this massive increase. This represents over time about 500,000 deaths. As of 2016, you see this precipitous increase, this blue line in deaths related to fentanyl. And that is on a local basis. The fentanyl epidemiology is very patchy. There are cities that go from almost nothing to, flooded with cases in a year or two. And as of 2016, New York City was leading the country in the rate of deaths from fentanyl. I have no idea where these figures are going to end up in 2017 or 2018, but they're not likely to be better. So what do we have right now? Why do we want to study a mechanism of action of a novel compound and looking for the development of new treatment? Well, look at how well existing treatment is working. For substitution therapy, buprenorphine or methadone maintenance, the retention in treatment, receiving substitution therapy, is about 5 months on the average. And then with regard to detoxification, if people want to detoxify and get out of maintenance treatment, or if they want to detoxify and get opioid free altogether, that is going to work only about 2/3 to 1/2 of the time. So approximately 1/3 to 1/2 of individuals who attempt detoxification by conventional tapers fail to complete treatment. So I think this also contradicts some statements that, oh, we don't need new ways for detox. We've got detoxification covered. We just, need a vaccine for cocaine or whatever. Detoxification is still problematic, and I don't think This audience needs to know that. I don't need to tell you that. Approximately 75 to 80% of those people who discontinue maintenance relapse the opioid use within 4 to 6 weeks. So that is what we have going in terms of present treatment. So now we're going to talk about mechanisms of action, and basically the point here that's going to be made, the overarching point, is that Ibogaine's mechanism of action is novel, and it is not explained by surface receptor interactions. It is explained by probably downstream post-receptor interactions, and that's sort of the point from here on out. Okay. So here are some theories of Ibogaine's mechanism of action in approximate order of appearance. I'm not going to go through all of them, and this is not even a complete list. There have been many of them. So let's start by distinguishing Ibogaines from some other psychoactive alkaloids. The Harmala alkaloids are kind of interesting. They, for example, enhance spatial learning in animals. Harmine-containing plants seem to be capable of spatial learning, like Passiflora. Ibogaine also enhances spatial learning, there are some structural similarities. And interestingly, harmine has some kind of an effect on diminishing opioid withdrawal, but it's not a very strong effect. And what it's acting as apparently is an imidazoline agonist, otherwise known as an alpha-2 agonist. For example, clonidine is an alpha-2 agonist, and it has some effect, but it's usually not clinically very consequential in people with high levels of addiction. The Harmala alkaloids are working in a different way. Also, the Harmala alkaloids are famously inhibitors of MAO and of monoamine oxidase. That's what they're doing in ayahuasca, and Ibogaine does not inhibit MAO. Okay, so classical hallucinogens, which are basically LSD, psilocybin, these are drugs that have a high specificity, and high activity as agonists at the serotonin type 2A receptor. There is no apparent effect of classical hallucinogens in opioid detoxification. And if you remove 90% of the RAF-A, which is the major serotonergic nucleus in the brain, it doesn't affect the expression of opioid withdrawal. And in discrimination studies where an animal is asked to identify the Ibogaine stimulus, the 5-2A receptor component is non-essential. The animal can recognize Ibogaine without it. It is also the case that with regard to Howard Lotsoff and his focus group, it is very likely that they ingested classical hallucinogens, as a matter of fact, certain, and they observed no serendipitous effect on opioid detoxification with the classical hallucinogens that they were using. And as far as kratom is concerned, kratom is an opioid agonist. Ibogaine, as we're going to see, is not. Okay, so a little basic concept in terms of the physical chemistry. Sequences of amino acids form proteins. Okay, and this is how a protein is often represented, and in silico graphic shorthand these days. So these are basically, these are spirals, bundles of helices formed by chains of amino acids. And the sequence of the amino acids determines structure, and structure determines function. What we're looking at here, for example, is the structure-function relationship of the two conformational states of the SERT, the serotonin transporter. Okay, and there is basically what you can see is it opens up out here, and then it flips and then releases in here. And perhaps a better graphic representation is this. It's open and extracellular.
Hello, Ken.
Yeah.
Okay, we lost you for about 20 seconds, 30 seconds.
Well, you know why? That was Jonathan Dickinson calling on the other line. Is that like the Puidi coincidence factor or what? Okay. At any rate, so I was talking about the serotonin transporter, right? The serotonin transporter is a really critical pharmacological structure because basically when we talk about serotonin reuptake inhibitors, the SRIs, the RI part, reuptake inhibition, stands for inhibiting the transporter, which Ibogaine does as well. It probably has nothing to do with its mechanism of action and opiate withdrawal, but it's kind of interesting to structural biologists because Ibogaine does it in an unusual way. So the serotonin transporter, first of all, we have extracellular space where it opens up and it grabs a molecule of serotonin and it's recycling it and bringing it into the cell. So it basically flips conformation. So you go from the outside open and then you go to the outside closed, and these are the two conformational states. All of this is accomplished through the movement of molecules in space. And that movement is fueled by energy, and that energy is ultimately initiated by the bonding of a drug with the surface of the SERV. And with the transporters in general. So basically, the basic concept here is that structure, a peptide sequence determines structure, and structure determines function. So we're talking about modification of the structural shape of proteins in terms of the proximal actions that these drugs do. Okay. All right. And then alkaloids themselves we're now going to talk about a natural product class called alkaloids. And it's very interesting because natural products, as we know them now, were initially called secondary metabolites. The term is still around, so somewhat. This is basically chemical detritus. Alkaloids is the byproduct of primary metabolic processes such as photosynthesis or energy metabolism. And so the original view of alkaloids was that, and there are 20 or 30,000 of them in nature. There's approximately 200,000 natural products have been described. Alkaloids comprise a significant fraction of them, 10 to 15% of those that we know. What are they doing in the plant? Initially, the thought was that they're just metabolic waste. You know, they're just basically waste products of the plant trying to grow and breathe and respirate and do what it does. And that view ultimately was abandoned in the previous century, in the 1900s, with the ecological view, which prevails today and is definitely valid, that alkaloids function as an ecological function to ward off predators or to attract pollinating insects. So it's basically an ecological view that alkaloids help the plant interact between the plant and its environment. What we're seeing now, though, and this is, I think, an emergent, picture that is going to influence drug development, is that alkaloids such as Ibogaine may be endogenous modulators within the plant itself. And given the fact that the plant may have, as we're going to see in subsequent slides, the plant has some structures in its signaling pathways that overlap with ours. The idea that an alkaloid may have a function within the plant itself is interesting in terms of a cross-species communality. This is something that we're just beginning to look at. There's been a few descriptions of alkaloids definitely playing a modulatory function within the plant and not just between the plant and its environment. Okay, so alkaloids by definition, what are they? There's no universal, agreed upon definition, but there is a large consensus as to what an alkaloid is. Generally, they're formed from the same pathways as amino acids, and they extend on amino acid structural scaffolds, notably with regard to the aromatic amino acids. And basically, this is why alkaloids are so avidly binding to proteins, because alkaloids themselves are derived from some of the basic building blocks that proteins are as well. An alkaloid contains a basic nitrogen. Here's one. Nitrogens donate electrons, and that helps them distribute charge in space and have the effects they have in terms of rearranging protein structure in space. So a nitrogen group and a ring system, this means that there's a lot of electrical charge that can be distributed in an unimaginable variety of ways. Basically, infinite alkaloids can distribute charge in space, and they do it with precision and with exquisite variety, essentially infinite variety, and often quite stably. So Another thing that's typical of ibogaines is diverse receptor binding and off-target effects. Many of these abused substances that we treat and study, heroin, cocaine, nicotine, amphetamine, the classical hallucinogens are all themselves alkaloids as well. So just looking at ibogaine, you can see this is classically an alkaloid. If there is an alkaloid country, its heartland, Kansas, would be Ibogaine. It's very, very representative of the class. Okay, and this is the Ibogaine has diverse affinities typical of an alkaloid. This is a rather dated diagram, but nonetheless, what it shows is that basically there's these are receptor affinities of Ibogaine, noribogaine, and 18-MC. And notice also, you know, the individual affinities are not of interest here. It's just basically showing that these alkaloids, that Ibogaine, typical of many alkaloids, has lots of different affinities. Not all of these affinities are important. Some of them are probably exerting a no effect or off-target effects, side effects only. And none of them may be involved in the therapeutic effect as opposed to more downstream structures. But notice that there is also a distinction between functional effects and binding affinities. Binding is just the strength to which a molecule binds to a receptor. It could bind to the receptor very strongly, but not initiate any kind of an event. I'll give you an example. The hERG channel, Ibogaine and 18-MC bind to it equally tightly. They are equally avid in terms of binding the receptor, but the functional consequences of binding in terms of blocking the hERG channel are much greater for Ibogaine than they are for 18-MC. So binding and the functional effect are different things. Okay, Ibogaine, alkaloids are a single class among classes of natural products. Indole alkaloids, which is the class Ibogaine belongs to, is a single class among multiple possibilities. And the indole ring, the indoles is what has been called a privileged scaffold. This is a term from medicinal chemistry. A privileged scaffold is a basic molecular framework or structural skeleton, defines a class of compounds with systemic modifications allow modulation of therapeutic or adverse effects. And that is, definitely the case with the iboga alkaloid scaffold. Alkaloids are formed in plants from pathways of synthesis of amino acids. Alkaloids, as well as neurotransmitters, extend on amino acid scaffolds, particularly with regard to aromatic amino acids such as tryptophan, which is an example of an aromatic amino acid, which is the precursor of serotonin, and it is also a precursor to the synthesis of ibogaine. Indole alkaloids are particularly well adapted structurally for interactions with proteins because they distribute charge in space in an interesting way. This is a charge diagram on the right of here we have the stick diagram of the indole scaffold, and this is what it looks like in terms of the electron cloud, the charge cloud, and you can see there is a big difference. You're looking at a gradient here where cold is low and hot is more charged, but basically the point here is that these shapes are capable of distributing charge in space in a variety of interesting and important ways. And this will determine their interaction with larger signaling molecules, which will activate or inactivate them. Okay, target structures include G protein coupled receptors and their effectors and related signaling proteins, and we're going to be hearing more about them. Okay, most of the classical hallucinogens, for example, as well as ibogaine, are indole alkaloids, and ibogaine is a special kind of indole alkaloid, a monoterpene indole alkaloid, which is a particularly important medicinal class. Okay, so signaling pathways. Okay, so nobody should be intimidated here. We're talking about a general point. There's going to be a lot of pictures with more information than you need to know. But basically, just concentrate on the flow of the visuals here, and I will explain the rest. So let's start with something really simple, the direction of signal flow. This is an animal cell, and, like a cell of somebody who's taken a drug, and it's got a nucleus, and it's got an outside external membrane, and the receptors are out here, and the nucleus is in here. The receptor senses the environment and transmits a signal to the nucleus, and the nucleus basically mounts a response to the environment. Proteins are synthesized in the nucleus. So if the signal flow comes in, it's getting cold, the nucleus will encode a protein that says grow more fur. Or if an opiate, if it's a powerful synthetic opiate is coming into the receptors, the nucleus will generate proteins to modify the sensitivity of these receptors to be able to survive this onslaught of opioid activity. So sensing at the surface, response in the nucleus. And so here we have a scheme of signal flow. And this is actually an old diagram. I think I got this off a NIDA website. It's at least 10 years old, but it has the basic signaling elements. So let's just concentrate on this over here. Okay, so the levels of signaling, the receptor outside the cell, the transducer crosses the membrane, the effector, the second messenger, the kinase, and transcription factor, and I'll explain what these are. Okay, and this is just the same flow represented with a different looking picture. So basically there's a directional flow here that is kind of a stable structural representation of all of these receptors that we're interested in, called G protein coupled receptors, which are, among other things, the receptors to which opiates bind, as well as dopamine and serotonin and muscarinic acetylcholine and a few others. Okay, so stages of signaling, let's describe them without all the pictures. The receptor senses the extracellular environment. Extracellular environment can be in a person's body, but external to their cells, for example, a brain cell, and then the brain is being exposed to heroin coming in through the blood-brain barrier. I'm getting an internet connection problem signal. Are you guys doing okay? Hello?
Yeah.
Oh, you're okay. All right. All right. So the transducer, this is important. The transducer converts diverse environmental stimuli and receptor interactions to a common signaling code. You have the receptor sitting on the surface of the cell. You have all kinds of different receptors for all kinds of different chemicals and ligands. And some of these receptors are receptors that sense sugar content in the blood, or sense the temperature outdoors. So basically, there's all this information coming in the form of either chemicals or conditions that needs to be translated into a common metric so that the signal can be processed. And that occurs with a transducer, and that occurs as the signal is crossing the membrane. Okay, so you have, for example, an opioid binding to a receptor on the outside, and then you have a transducer transforming that into a coded event that starts off an effector, and the effector is sitting inside the membrane, and that originates the downstream intracellular flow that's going to follow from that. Effectors produce second messengers, and that's how their flow is propagated, and then that reaches kinases. Kinases are the barcode. They're situated between the effector and between the nucleus, and basically, That looks like Jeremy's.
This dropped.
What dropped?
The presenter. Jeremy, can you hear us?
Yeah, fine. It's all good. It's all good.
Okay.
Are you seeing the screen? Okay.
Oh, okay, we're getting an error message here, but you can hear me? Okay.
Yeah, we've lost the presentation.
We lost the presentation.
That's right. Back to you. Share screen. Are we back?
Are we back?
Not yet.
How about now?
Coming up. Not
Oh, you
yet.
just.
Hasn't come up on the screen yet. Yep, we're back. We're back.
Okay. All right. So again, so we're talking about the receptor sensing the extracellular environment, the transducer across the membrane. The effector is now taking hearing the signal and transmitting it downwards. It's transmitting it downwards to a kinase. Now, what's interesting here, the kinase is the barcode, meaning that it's putting a unique stamp on constellations of events that are going to occur all at once. So it's regulating upstream and downstream, kind of connected to everything, producing a unique response that basically it phosphorylates. It puts phosphate groups on molecules, and by doing so, it activates or inactivates them. And those, you can think of those phosphate groups on molecules as individual yes/nos, digital bits of information. And if you add a bunch of those together, you have a unique barcode. And basically, in this region, in this region, you have responses that are very specific ensembles of responses. This is what we do when we want to grow fur when winter comes. This is what happens when we have to make neuroadaptations to the administration of large doses of opiates and so on. So these are unique responses that are encoded. But basically, in terms of pharmacological targets, the G protein that are not receptors, modulators of G proteins and modulators of effectors are prime candidates, and also certain kinases. And we'll get into why these are strategies that might be expected to be useful in terms of drug development. Okay, so again, the same scheme applied to the same to a different figure. Okay. Wait, am I going? Okay. So we're going to talk about one signaling pathway as a prototypic example of the larger phenomena of GPCR signaling. So let's start with opioids. Why not? So the receptor in this case is the mu-opioid receptor. The transducer is a G protein. You've heard the term is G protein coupled receptor, or GPCR. It means a receptor such as the mu-opioid receptor. It is coupled to a G protein, and we'll talk about what that means. The effector is something called adenylate cyclase, and these are the elements of the signaling pathway. In other words, these are the general elements in a schematic signaling pathway, and these are the specific elements that correspond to the opioid signaling pathway. Okay, so let's talk about at the receptor level. Okay, first of all, there are multiple kinds of opioid receptors. There's delta, mu, and kappa. And these are, again, in silico pictures, representations of the three kinds of opioid receptors, and the pockets in where the drug binds are represented in here. And this is delta, mu, and kappa. Withdrawal is mainly encoded by delta and mu and not kappa. Kappa encodes actually aversion. It is increased in withdrawal. So these are some of the functional differences among the receptor subtypes. And the receptor of greatest interest as far as administered opioids is concerned, morphine, heroin, fentanyl, and so on, is the mu-opioid receptor. There are endogenous opioids in your body. Hello? Yep. Are there endogenous opioids in your body? And beta endorphin is an example of one of them. That's a familiar term. Endorphin is something that the body makes that binds to the mu-opioid receptor. So basically, the mu-opioid receptor has its own cognate ligand, which is the beta endorphin, and synthetic opioids bind to this receptor, just like cannabinoids bind to the cannabinoid receptor, and anandamide is the cognate endogenous ligand there. As one can see, there's not only in beta endorphin, but there's a whole bunch of other internally generated molecules that bind to the mu opioid receptor, the delta opioid receptor, and the kappa opioid receptor. A lot more than just endorphins. Okay. So the mechanism of action of Ibogaine is apparently unknown and, importantly, not explained by surface receptor binding. And we know this for several reasons. First of all, Ibogaine dosages that are used for detoxification in the setting of severe physical dependence don't produce overdose in opioid-naive individuals. Ibogaine nor Ibogaine 18-MC do not, in a assay of functional effects, it's called the gamma GTP assay. This is an assay that measures not binding, but whether there is activity. So there's a binding event that is initiated by an opioid binding to an opioid receptor, and that event is captured by this test. And so this is what Damgo is, a very powerful opioid agonist, a standard. And this is the activation that you see close to 100% with Damgo, okay? This is Damgo combined with either noribogaine, ibogaine, or 18-MC. And as you can see, at larger doses, these iboga alkaloids are acting as weak antagonists at huge concentrations. These are not physiological concentrations, but the point is there's no point here where ibogaine is an agonist. And actually, this is what a real agonist looks like, and this is what these compounds look like if you just add them by themselves. There's no activity that's initiated by these compounds. You're measuring opioid activation here. This is a standard opiate, and this is what ibogaine and 18-MC and noribogaine are doing, no opioid activity. And as we mentioned, ibogaine has no significant affinity for the imidazoline site, so it's not acting like clonidine. It's not acting like an NMDA receptor antagonist either, because 18-MC, which is a synthetic analog of ibogaine and is just as effective in the animal model and models of withdrawal. And the animal model generally closely reflects the human model, regardless of issues regarding validity of animal models and so on. With regard to ibogaine, there is a strong fidelity between animal models and human effects. 18-MC has no effect has no effect on the NMDA receptor, and yet has similar actions in suppressing opioid withdrawal signs in the animal models. So that rules out the NMDA receptor, which was once a leading theory as to how ibogaine works. There's also the alpha-3 beta-4 nicotinic receptor. We have antagonists at which ibogaine and 18-MC are agonists at the alpha-3 beta-4 receptor, and there is some effect of the alpha-3 beta-4 on opioid withdrawal, but the question is, how significant is this? It's an in vitro effect. There's a lot of things that have small effects on opioid withdrawal in some cellular model or a mouse model, but the question is, do they have the kind of effect that ibogaine has of detoxifying somebody from a half a gram or a gram a day of heroin or the equivalent? Interestingly, we have in bupropion, otherwise known as Wellbutrin, a nicotinic antagonist, and it is a nicotinic antagonist at the alpha-3 beta-4 receptor. And I know of no severe clinically important interactions of bupropion with opiates, which would surely be the case if bupropion were suppressing opiate withdrawal and modifying, sensitivity opiates as ibogaine does. Ibogaine and 18-MC and bupropion all bind to the alpha-3 beta-4 receptor. So the alpha-3 beta-4 on its own doesn't seem to be explanatory. Okay, my favorite theory, the surface receptor ensemble theory. Since we're at the receptor level, I'm throwing it in here. As seen as Wikipedia, this is really annoying to me. The multiple surface receptor ensemble theory holds that there is a precise, complex, specific, but entirely unknown polypharmaceutical constellation of surface receptor interactions that mediates the clinical effect. In other words, it's entirely post hoc. We think that because ibogaine has multiple receptor interactions, that those interactions must somehow be important and mediate the clinical effect, which means that you can state this even if you don't know anything else. Very convenient. It's in Wikipedia, which is, of course, the arbiter of scientific truth, and it carries a low burden of proof because it's exempt from the requirement of experimental confirmation. Because if somebody were to do an experiment and say, okay, these are Ibogaine's receptor interactions, let's systematically vary them and see if we get a dramatic effect on opioid withdrawal. That's not likely to happen in terms of happening on the exact one, especially if it doesn't exist. Then you're always going to get negative results, but you can't prove the negative. The person advancing the theory has the advantage of saying, well, there's some combination, you just haven't found it yet. Against this argument, the theory relies on the conviction that a theorized specific polypharmaceutical combination should exist. This is entirely post hoc. There's never been any attempt to state the combination of actions that should comprise the profile. Now, what about the fact that Ibogaine has so many interactions that it's a dirty drug? So what? Alkaloids typically bind to multiple diverse receptors. That's what they do much more often than not. And a lot of those binding affinities are unrelated to the therapeutic action. So if you're going to attribute essentialness to a receptor interaction on a post hoc basis for an alkaloid, the statistical chances that any individual attribution is more likely to be wrong. 18MC is an interesting example in terms of studying mechanism of action because it's kind of a control comparison to Ibogaine. It does not have interactions with the SERT, with NMDA, or the sigma-2 receptor, which means that those structures probably can be ruled out as essential to Ibogaine's mechanism of action. By the way, the multiple surface receptor ensemble theory, it dates back to a different era. This was in some of the early reviews, and that's, I guess, why it lives on in Wikipedia, and was in the early 1990s is when it originated. That's when publication volume on Ibogaine began to pick up with the NIDA Ibogaine project and the work that immediately preceded it. In those days, a role of post-receptor signaling was not yet appreciated. Everything was thought to be receptor mediated. We're talking about a generation ago. And interestingly, Eric Nestler, who is probably the most highly regarded neurobiologist studying addiction these days, turned in a grant application in the late 1980s, and he received this comment from the reviewer. There is no evidence to suggest that post-receptor signaling is important for opiate addiction. So this is the era of the surface ensemble theory. And Nestler eventually did get the grant and subsequently has done very well as a neuroscientist studying post-receptor mechanisms in addiction models. And he is not known to cite Wikipedia. Okay, so the transducer is the next step. The transducer in this case is something called the G protein. And the G protein goes through a cycle. Here's the receptor, and the G protein is what is called a heteromer. It is composed of 3 proteins. There's an alpha unit.
Sorry, we're stuck on the previous slide, Ken. We're still on the Eric Nestler slide.
Wait, connecting again. Let me know when we make the shift, because you apparently are able to see stuff before us.
You're seeing the preview on your phones.
I'm seeing the audience.
Now you are. Oh, yeah. Yay. All right.
Unmute. Yep.
Wait, share. PowerPoint.
Okay. Are we back?
Nearly. Yeah,
Right.
back. We're back.
We're back. All right. Okay. So G proteins, the transducer stage. Okay. At rest, you've got the receptor. It's linked to a G protein, coupled to a G protein. So this is the receptor. These are the G proteins. And this is a G protein coupled receptor, coupled to G proteins. And the G proteins undergo conformational changes when a receptor binds. So if this is a mu-opioid receptor, we have morphine binding here and activating the G proteins. the G protein becomes activated and separates into an alpha component and a beta gamma component. Both of these components are capable of signaling streams. And there is something called signal biasing. We're not going to get into it, but just to, if you've heard the word, signal biasing has to do with the relative amounts of bias of the G protein signal versus the signal that is originated by the beta gamma portion, which involves a compound called beta arrestin. Basically, this has been a big hot topic. It has not been highly predictive, though. Biasing has not been highly predictive in terms of drug development, because there's not only two choices here of either beta gamma biased or alpha biased. There are hundreds. This is an oversimplification. But at any rate, so that's why I'm not going to get real into biasing, even though there's a lot of talk about it. But at any rate, downstream, you get what are called effectors, and that's going to be in subsequent slides. So you go from inactive to active, and then it cycles around. Okay, so receptor neurotransmitter binding causes splitting up of the trimer, of the heteromer. Once it's split up, the active forms initiate downstream signaling. And then once the receptor occupancy, once the drug leaves the receptor, then it goes back to where it was.
You dropped again. So there's no slide advance.
Mm-hmm.
They're gonna.
Why do you think that's happening?
No, we can see the slide.
What? Okay.
We can see the slide.
Okay.
You want to?
Now we can't.
Oh, okay. Thank you. So what we did before was we attempted to reconnect.
Mm-hmm.
Let's see.
So you think it's at this machine's end?
Yeah.
Really?
So now when this pops up,
Mm-hmm.
we share. Go to PowerPoint, and now we're back.
Okay, it's coming back.
Okay.
All right,
right.
it's back.
So again, showing the transducer, the GPCR. This is the receptor. This is the G protein. The G protein undergoes these conformational changes, and then the activated signaling elements initiate downstream transmission. Conceptually, the main thing here is that at the receptor can be sensing temperature or sugar content or any number of chemical neurotransmitters or drugs. And this transducer stage is translating them into a common code. And that's the main thing to know about the transduction stage. But this is also, there are certainly proteins that the body generates that modulate this G protein signal, which is another a drug target that could be downstream. Now, interestingly about G proteins, the last universal common ancestor had G proteins. These are phylogenetically ancient signaling elements, so they're a good candidate for something that is a target both within the plant and us, which is what I suspect may be at the root of Ibogaine's mechanism of action. Plants and animals evolved from a last universal common ancestor. That's a thing. The last universal common ancestor, LUCA, is an organism that was the progenitor to both plants and animals before we went our separate evolutionary ways, which would be about a billion and a half years ago. G proteins in animals and plants are very similar. They are strongly conserved, meaning that the amino acid content it matches an up to fit, which is called homology. That homology is up to 50% for G protein. So this is a really remarkable stage of signaling that seems to be universal among plant and animal life. Plants and animals, what do we have in common? Well, we're both eukaryotes, which means we're cells with a nucleus. This is an animal cell. This is a plant cell.
We're frozen, and that signal is coming forward to the next slide. Jeremy, are you hearing us?
Yeah, but I think it's frozen again.
Yeah.
Did it unfreeze yet?
No, it's black.
Oh.
So what we do is we attempt to connect through the browser or another thing. When it does, then we can say that that's pretty good. Share. Touch that.
I don't see my. Oh.
Okay.
Mm-hmm.
So that's the camera phone.
Mm-hmm.
So you have to get them in here.
Mm-hmm.
And of course it's
Looking for my mic or?
this is the mouse.
Oh, yeah.
There she is.
Mm-hmm.
Share. PowerPoint.
Okay.
Okay, it's
Watch this
coming
come
back.
up.
All right.
Taking a while to come.
We're just waiting for it to
Okay, it's
sequence.
back.
There you go.
Okay. So plants and animals are eukaryotes, meaning that we have a well-defined cell nucleus. So we have a need to communicate from a cell surface to a nucleus. Interestingly, plants do not have G protein coupled receptors. At the surface, plants have G proteins that are sensing, temperature and sugar concentration conditions and so on, but not through receptors. Receptors is an animal thing, not a plant thing. But we both have G proteins. So let's look here at signaling pathways that occur in plants and that occur in animals, and which which ones do and which ones don't, for two signaling pathways: opioids and classical hallucinogens. Okay, so if it's red, it's only in animals. If it's green, it's in plants and animals. So notice that plants do not have receptors, we do. And G proteins, which is the signaling element, the transducer, we have and they do. So this is also a likely modulator. This is also something that can be modulated in a drug that is that moderates withdrawal and opiate effects. Downstream from the G protein, you have the effector. And the G protein is acting on the effector. It's either stimulating it or suppressing it. And there's one effect. So here we are. We're right in here. The second messenger is manufacturing an effector. Okay, it is being modulated by the G protein. The G protein can tell the effector what to do. Now, this is interesting because we have another candidate here where there is some experimental evidence that Ibogaine's mechanism of action could be involved, which is at the effector level. Okay, and this is an experimental model of opiate withdrawal that I think most opiate pharmacologists would agree is the leading cellular model of withdrawal. It was developed in the 1970s, and basically, if you chronically give opiates, it will suppress an effector called adenylate cyclase. And adenylate cyclase makes a signaling molecule called cyclic AMP. And morphine inhibits it. If you give a cell culture morphine and you get it dependent, which you can do, you give it morphine, it gets dependent, it gets tolerant to it. And then you suddenly take it away by administering naloxone. The cells themselves will go into withdrawal, and they will manifest this by a surge in cyclic AMP. So here's cyclic AMP, and here is the administration of naloxone induced withdrawal. And you can see all of a sudden, there's a huge surge in the production of cyclic AMP, which is a product of adenylate cyclase, which is the effector. And adenylate cyclase is regulated by the G protein, and the G protein is regulated by the G protein coupled receptor, the opioid receptor. So if you can generate signals that could otherwise have come from the opioid receptor at the level of the G protein or at the level of the effector. So basically, you can suppress the signs of withdrawal by suppressing adenylate cyclase and not giving an opiate. And that is a conceptually opening the door to a possibility of a drug target that is not a receptor that is effective in opiate withdrawal. And there is, in fact, some evidence that adenylate cyclase is a good candidate. In the rat, there's different types of adenylate cyclase, which we abbreviate AC. Adenylate cyclase is increased. It's like a compensatory adjustment. It's an equilibrium type of homeostatic adjustment. If you're giving opiates, it's going to suppress the activity of adenylate cyclase. The body has to fight to regain equilibrium. It has to produce more adenylate cyclase, which is being suppressed by the opiates. And it fights back by making more adenylate cyclase. This has been demonstrated in the rat. You give the rat chronic morphine, and after a while, you have increased expression of adenylate cyclase as a compensation for all the suppression of adenylate cyclase that the opiate has been causing. So adenylate cyclase being overactive and producing too much cyclic AMP is withdrawal. And adenylate cyclase being quiet is satiation and the resolution of withdrawal symptoms. So what if you had a medication that operated directly on adenylate cyclase? Or what if you had a medication that operated at the G protein level, which is inhibiting adenylate cyclase? Again, these are drug targets that are possible that are not surface receptors. And in fact, a small molecule inhibitor of adenylate cyclase produces analgesia, which is a cardinal mu-opioid receptor mediated symptoms. Also, knockout mice, in other words, mice that have no adenylate cyclase, exhibit reduced opioid withdrawal signs, which you would figure if AC adenylate cyclase was involved in the production of opioid withdrawal signs. And AC on itself may be a target for the development of pharmacotherapy of opioid and addiction. And likely, if it's pharmacotherapy for opioid addiction, it is effective for other drugs as well. And that would include Ibogaine. So basically, is there any specific evidence regarding, we have conceptual evidence regarding adenylate cyclase and opiates. What about Ibogaine adenylate cyclase? There's some very old work that's been done on this. And we're talking about the mid to late 90s. And some very interesting work. And I asked the person who did this work, because I think this is some of the most important work that's been done in the area, why he did not follow up on this. And he told me that he was told that working on Ibogaine back in those days was not a good career move. Anyway, so this very provocative finding was published quietly in, I think, 1996, that Ibogaine potentiates the inhibition of adenylate cyclase, but morphine inhibits adenylate cyclase. That's what it does. And Ibogaine potentiates that inhibition. That's the potentiation of an opioid-like effect without adding opioids. That's quieting adenylate cyclase down which is mediating with adenylate cyclase mediates a withdrawal state. So if you go into withdrawal, adenylate cyclase is freaking out. It's producing too much adenylate cyclase. You're going to quiet that down with exogenous opiates. You're going to quiet that down with a suppressor of adenylate cyclase. And apparently, opiates which suppress adenylate cyclase do it more when they're treated with Ibogaine. So this potentiation of the inhibition of adenylate cyclase is consistent with an effect in the opioid detoxification. And this is an important lead that should have been followed up and hasn't, and I'm working on that. Anyway, so the kinases, the barcode, the main thing here, kinases are incredibly complicated. There's a lot of them. They too are drug targets. I'm not going to get into that.
I think we're stuck.
Enough of that.
Okay.
We're towards the end here. Basically, the thing to notice about the kinases is that the arrows go in both directions. These are regulatory proteins that are phosphorylating downstream and upstream to capture and barcode a specific multi-faceted response. Okay, and then regulators of gene expression, last stop, is basically genes are protein. Genes are coding for proteins, and proteins can suppress adenylate cyclase, and proteins can regulate G protein coupled receptors, and proteins can phosphorylate receptors and change their activity. So all of this response to administered opioids can take place in the nucleus. Here's an incredibly busy slide, and I just want you to look at one thing. What we're having, this is basically genetic coding. You got your double helix here, and it's a plan, it's a blueprint for the production of proteins. And transcription factors will modulate the production of proteins. So basically, at this last stage in the nucleus, the transcription factors are regulating the response to proteins. And this is, by the way, where Nestler did all the work that he's recognized for that was initially rejected by the NIDA reviewer. That concludes the scheme of receptor, transducer, second messenger, kinase, and regulators of gene expression. We've got the GDNF story. GDNF is a neurotrophic factor. These are growth factors. They mediate plasticity. They're also involved in, and they're promoted by plasticity is also promoted by serotonin. GDNF is a potent neurotrophic factor. It's been identified affects the midbrain dopamine neurons, and Parkinson's disease is a loss of dopamine neurons. So the logic that GDNF might be helpful in Parkinson's disease has attracted a lot of interest. This is a paper where GDNF, reporting on the results of GDNF administered through an intracerebroventricular catheter. In other words, a tube through the skull into the brain, directly into the brain, which indicates the level of interest in getting GDNF into the brain to treat Parkinson's. This paper has, by the way, been cited over 800 times. And Ibogaine enhances the expression of GDNF. It may do it through a positive feedback loop, which makes sense for a growth factor to induce its own expression. But there is a confound here that needs to be addressed before we get too excited about this idea, which is basically this. We talked about how Ibogaine binds to the SERT, and compounds that bind to the SERT include antidepressants. And they also, in cell culture, will enhance the expression of GDNF. So here we have a table of a bunch of things that enhance the expression of GDNF in cell culture. Ibogaine is just one of them, and the magnitude of the induction is similar among Ibogaine, let's say, and antidepressants. The enhancement of Ibogaine may be an you can be a devil's advocate and say that the enhancement of GDNF expression is nothing but a product of the blocking of the SERT, just like a Prozac type effect. But you can't detoxify junkies with Prozac. This raises the possibility the effect of Ibogaine, Norbogaine, or GDNF expression may be an epiphenomena of both compounds blocking the SERT. But you've got 18MC, which has no significant affinity for the SERT. So what does 18MC do? Well, in cell culture, Ibogaine and 18MC differ. Ibogaine enhances the expression of GDNF in most cell cultures. There is actually some negative data that has never been published, but in most of the time, Ibogaine enhances the expression of GDNF. 18MC does not in cell culture, but it hasn't been looked at in the intact animal. Why is that important? Because when Ibogaine is given systemically, in other words, you give it orally or you give it intraperitoneally, which is how it's usually given, you get a GDNF expression effect, but that hasn't been done with 18MC. So if you give 18MC to an animal and it causes the expression of GDNF, that's a very interesting finding, because you know then that it's not an epiphenomenon of its interaction from the SERT. You know that that has something to do with an Ibogaine, or it's something to do with the basic structure. Okay, so that's an interesting result of Ibogaine if 18MC administered systemically causes the expression of GDNF. And if it doesn't, it raises a question as to whether the production of GDNF is important in Ibogaine's anti-addictive mechanism of action. After all, if 18-MC does not cause the expression of GDNF and Ibogaine does, or both are equally effective, then that raises a question as to whether GDNF is a factor in the anti-addictive properties of Ibogaine. So this experiment of administering 18-MC to an intact animal really needs to be done. And that brings us to the end of this presentation. And I just like this quote: almost more than talent, you need tenacity and an infinite capacity for rejection if you are to succeed, which I think is really good advice for somebody who wants to study the mechanism of action of Ibogaine. Thank you.
Have you got time for questions, Ken?
Yeah.
Okay, questions. Dana, you must have a question. Ah. Well, no, we have another question first. Okay. Dana Seeds. Amazing.
Hi, Dr. Ken. Great talk. Thank you very much.
Who is this?
Dr. Fred. I'm a Portuguese doctor.
Oh, yeah. I've met you. How are you doing?
Yeah.
Nice to meet you.
Nice to meet you. Yeah. Thanks for the talk.
We've spoken on the phone, haven't we?
Yeah, we have. Yeah.
Okay. Right. I still can't see you. You're off camera, but
yeah.
Anyway, I'll look forward to figuring out who you are.
The technical crew wishes not to show my attire today, so.
Oh, there you are. Okay.
Yeah. So I would like to ask something which I think you haven't addressed, but probably you might have thought about it before. What are the future prospects of dealing with the ERG blockade? Because I think that's a very pressing matter.
Well, I think that there's two fronts on which you can pursue. One is to look at compounds that don't block HERG. 18-MC is one of them. As I mentioned, 18-MC binds to the HERG channel with the same strength that Ibogaine, but does not functionally cause the same degree of blockade. 18-MC at this point in time is ready to be given humans in this country. And I think because of the HERG issue in terms of a regulatory sense, the HERG issue came into its own in the mid-90s. That was after Ibogaine already been approved in a clinical trial. So the regulatory climate of today is simply not one that's likely to accept the use of Ibogaine. Now, we can all argue, but what about the risk-benefit and, whether you think it's justified or not? That is a regulatory reality. Now, I think that one approach to this would be to do high-quality studies and possibly in a EU country like Spain or possibly Portugal, and do very high-quality studies, well-controlled and so on, and demonstrate that the drug can be used safely in a hospital setting, which I think with appropriate monitoring, it probably can. And I think also you have to look at the risk-benefit ratio. Methadone is one of the major causes of torsades. Most torsades cases, the two drugs most strongly associated with torsades in this country right now are an antiarrhythmic called amiodarone and an addiction treatment called methadone. So these treatments are not without risk. But I think the way forward is to do high quality, with regard to Ibogaine, is to do high quality studies outside of this country to demonstrate effectiveness and demonstrate the ability and develop protocols to administer safely. Admittedly, if the FDA comes around tomorrow and says, okay, we want to do a study on Ibogaine, what's the protocol? What is this protocol for safety? That would be an issue that would be elicit a lot of debate in the Ibogaine community, for which I don't think that there is a ready, glib consensus. So those are the fronts I think on which we can move.
Okay, a small second point was, have you ever thought about the possibility of spinal fluids, injection of Ibogaine, alkaline?
Well, that's been done in animals. And you would do that if you have any doubt that Ibogaine is getting into the central nervous system, but its central nervous system penetrance is apparently quite large. I'm thinking of my experience studying autopsy cases in the Ibogaine deaths. And there were a couple of those cases in which they measured brain levels of Ibogaine and noribogaine, and they were really high. It concentrates in the brain, gets into the central nervous system when it's given orally. So there's apparently no need to administer it through the cerebrospinal fluid.
Yeah, the point would not be the concentration, but the avoiding the herd blockade. That's the deal, right?
Oh, I see what you're saying. Well, yeah, that could possibly do it. There's a number of other, safety issues that would go along with that. I don't know that has been brought up. The question is, so you're going to implant a catheter for what?
Long term,
I guess
even
Parkinson's. I guess
Parkinson's
for the duration of the
patients,
treatment.
Yeah.
Yeah.
Long term.
I guess there is animal work to back up such an idea. The originally, this was back in the days in the Netherlands. They were very short on Ibogaine and actually administered it ICV, intracerebroventricularly through catheter in rats because they didn't have enough to give it systemically. So it certainly works in that way.
It came back. I want to
Hello?
ask, it's Dana. I want to ask you a question, Ken.
Hi, Dana.
We've been talking today about relative microdosing of Ibogaine for Parkinson's.
Yeah.
And I'm going to Prague to see about microdosing for crystal meth.
Yeah.
Because, long-term effects of crystal meth include Parkinsonianism. What do you think the problem with herd blockade is going to be at the microdose? Is it safe? Can we go ahead with microdose?
Well, what I've seen, I can't comment on whether it's safe or not. Interestingly, from what I know about patient D and others, there have not been dramatic effects on the QT in this dosage range. It may be the low dosage. It may be the small number of patients, because basically you don't see EKG changes in everybody. But you see it in enough that there is, that this is a very significant safety concern. So far we haven't seen this, but this needs to be studied systematically. Obviously, frequent, intensive EKG monitoring with special attention to abnormal waveforms and QT length would be a part of any study. Regarding Parkinson's and regarding methamphetamine addiction, the likely, I still think, regardless of my experimental reservations and my caveats, the GDNF model is the most compelling available model. And the enhancement of GDNF expression in methamphetamine would help counteract the obvious damage to dopamine transporters and dopamine neurons that's involved with methamphetamine. And there is some commonality between, you can look at methamphetamine as a degenerative disorder of dopamine neurons in a sense. So there's a logic there. The question is whether Ibogaine is enhancing the expression of GDNF by a trivial mechanism that is shared among other agents that have no effects in Parkinson's, or whether it is causing GDNF expression by some other iboga alkaloid mediated pathway, perhaps originating outside of the brain and projecting to the brain, in which case you wouldn't see it in culture. You'd only see it in the intact animal.
Follow up. We we got to go to. I'm going to let this pass to somebody else here.
I've got about a half hour.
Hey, Ken, it's Rachel Martin. How you doing?
Hi.
Hi. Thank you for that. It's always so interesting to hear your work, and I want to say thank you for doing this work for so long.
Thank
Really
you.
means a lot to all of us in the community. Another topic, two topics we've been trying to get to is aftercare, and also talking about a political way forward. Do you have any thoughts on that? Those two.
Well, they really aftercare is essential, and it's interesting. The conventional modalities keep resurfacing. Dimitri Mugianis, for example, went to Africa and so on and so on. But one thing he did after his Ibogaine treatment, since that got him off of opiates. After that, he went to a Greek island. He's Greek, and his family has access to an island where he spent 3 months without any access to anything, including dope, and then intensively worked the 12 steps for a number of years. And this is, people need to separate from their drug-centered lives, and if they're going to maintain recovery. There's a common designation of Ibogaine as providing a window of opportunity of reduced craving, which I think typically lasts from about 1 to 3 months, when the cravings are greatly diminished and people have the opportunity to reorient their lives. And if they don't, they're likely to relapse.
And
What was the other? You said aftercare. And what
so
else?
Political advocacy. Like, I think Dana would want to argue that it should just be completely unscheduled, and I tend to be more safety oriented and think it should be Schedule 2, the way MAPS approached MDMA.
Well,
research,
I would point out that, scheduling has not, it would be nice, but you can you get a Schedule 1 license, you put Ibogaine on it. It's not impossible. People have done it. I've worked in labs, obviously, where that's happened. There is psilocybin's a Schedule 1 substance. It is now being looked at here at NYU in a clinical trial for alcoholism. So I'm not sure that scheduling alone is, in itself, not going to do a whole lot. I don't think it's likely that because there's the issue here is, I think that you have to look at what the FDA does. The FDA designates whether drugs are safe or effective. And if you have a compound that is which safety and efficacy have not been adjudicated by the FDA, they can still make it illegal. I don't think you can, by legislative caveat, overrule the function of the FDA. I don't think that works legislatively. I don't think you can make a law that says, we want the FDA to suspend its usual critical process for this compound. What's the precedent for that? So at any rate, I think, at the end of the day, what you have to do, the FDA has never been the problem.
I'm going to pass this to, pass this to Dr. Gira, but yeah, I just have one third political idea, which is that maybe all the suits against big pharma, that they could somehow be brought into settling, supporting Ibogaine as an alternative treatment.
Yeah, it would be great if it goes to drug development. If it's in the billions of dollars, we're talking about the annual budget of NIDA. Keep in mind that the cost of bringing a single drug to market, and Ibogaine would likely be a fairly expensive project. Right now, the estimates for the pharmaceutical industry, the cost of bringing a new drug to market are between $1 and $4 billion. $1 billion is the low end. Now, there's different ways to calculate it, maybe opportunity costs or this or that, but basically you're talking about an incredibly expensive process. It's very risky, and right now, there's a problem with innovative compounds. It's not just limited to pharmacotherapy of addiction. This is, I think, a problem in psychopharmacology in general. I'm supervising residents who are using the same drugs. I'm a generation away from my residency. I'm supervising residents now that are basically using the same medications that were introduced when I was a resident. And the reasons why the pipelines have slowed down so much is that there's incredible risk associated with trying to develop these drugs. You put $600 million into it and all of a sudden, it fails, and that's that. Big Pharma does not like that. So basically, they take the regulatory paths that are well-traveled, that are low risk, and they basically develop compounds that are very similar to compounds that have already been approved. For example, that's why we have SRI antidepressants. We have now 10 of them, 10 that have been approved. We don't need 10. We need a new medication to treat addiction, but that is a very expensive development process. And if it's going to come from suits against Purdue Pharma, that's great.
Hi, Ken.
Hi.
I think when you spoke about scheduling, we were very happy that when New Zealand and then Brazil and then South Africa has decided to schedule Ibogaine, thinking that, we will be able to get it more acceptable. But from my experience, what I found out that the idea of scheduling was like an indirect way of telling us that, yes, we know it works, but we don't want to make it easy for you to use it. Because scheduling involves having certain conditions that's to be met. And it, at the same time, allowed for a number of underground service providers or Ibogaine treatment centers to continue without that so-called safety network. And especially when something goes wrong, then the full might of the law comes on to those using the so-called scheduling status. That's one. Number two, the other thing is that how does a regulatory body schedules a product or a medicine without doing any proper clinical trials, number one. Number two, also to schedule it to the level of the highest scheduling status with the same level as your morphine and maybe methadone, those type of so-called pharmaceutical accepted.
Well, it's actually more prohibitive than those.
Yeah,
But
so they put in the same category to the highest.
Well, yeah, but let's. I think we need to distinguish descheduling from approval. Descheduling would be, from a research standpoint, descheduling would be convenient. It would be easier, but a lot of Ibogaine research, ask Stan Glick or Henry Surgeon or other people that have studied Ibogaine, and they got a Schedule I license and added Ibogaine to it. Takes about a year and a half, but when it's done, it's done. There's lots of labs that are able to do research on Ibogaine. It would be much easier if it weren't scheduled. But descheduling doesn't mean approval. It doesn't mean that you can give it as treatment and call it safe and effective. The FDA would have big issues with that, and I don't think you're going to get a legislative fiat to overrule it.
Hey, Ken.
Yeah.
Go back to the issue about the herd blockade and deaths and stuff.
Yeah.
Because we know in Canada, they weren't. It wasn't even on their radar until they started making Ibo Plus, and then Health Canada said, if you're giving this in a really, really low dose, because of high dose effects that only occur if you give, the heroin-sized dose, we're going to ban it completely and come down on Bob Sisco like a ton of bricks.
Yeah.
So the question I have goes to this thing that's in Wikipedia again. The one in 300 deaths, that out of 300 Ibogaine treatments, one person dies, automatically, no matter what you do. And I have, is this based on a misreading of your work? Because we had about 1 in 425 deaths in.
Well, Dane, I don't think the number 1 in 300 or 1 in 400 is really that different.
Well, no, it is, because
given the level
methadone
of
is 1 in
uncertainty with the
372. So if it's 1 in 425, it's safer than methadone. If it's 1 in 300, it's a whole lot more dangerous than methadone.
Well, I'm not, and I don't know how you can say that the true number is either one of those. In other words, there is some uncertainty associated with the estimate. Don't forget in the original Ibogaine subculture paper, we estimated that something like 20 or 30% of treatments had been overlooked by our study. Similarly, deaths may have been overlooked as well. I don't know what the ratio is.
Well, where do they
I
come
would
up with
say
1 in
probably
300?
there's a range that you estimate. And whether it's safer than methadone or not would be a statistical comparison that would probably be rendered non-significant by the high variance of both terms so that your comparison. In other words, if you have a term that's estimated with a lot of variability and you compare it to another term estimated with a lot of variability, it's much harder to get a significant difference.
Okay.
But,
Okay, Ken, we have to finish there. We've got Doug on next, so thanks very much.
okay. Thank you.
Okay, 5-minute break, and then we have Doug.